So, hi everyone. Thank you so much for being here. I'm Yannick Francioli. I am in the Casto Lab in the University of Texas at Arlington. And I'm really excited today to show you some of our work on extending the genomic coupling theory on the empirical rattlesnake hybrid zone. So I'll start by describing the fundamental questions that we want to address here. So what are the mechanisms that generate and maintain species? How can we objectively characterize the speciation process and the progress towards it? And are there some processes that are required for completing this speciation process? And genomic coupling theory is really an attractive way to address those questions. It was fundamentally based on multi-client theory proposed by Barton in 1983. And I want to give a big shout out to Butlin and Smadja and their collaborator that did a tremendous amount of work on this subject and greatly inspired us to do that work. So now I'm going to try to do a quick introduction to coupling, which is kind of a challenge, but let's give it a go. So as lineage diverge in allopatry in this case, reproductive isolation increase through accumulation of barrier traits, and upon secondary contact, barrier traits and the underlying loci, they reduce gene flow between those lineages. And so coupling, genomic coupling is really fundamentally driven by this tension between recombination and selection to keep parental alleles together. That's Barton's proposed coupling coefficient, S over R, which means selection over recombination. And what that means basically is that hybrids that do not have the right combinations of parental alleles will have a reduced fitness, and that will give rise to patterns in the genome of hybrids in the hybrid zone. And so that leads to association of barrier traits and underlying loci. That leads also to buildup of LD, as Eric Dorpman just discussed before. And cumulatively, these barrier loci will represent a stronger barrier to reproduction together. Going a bit further into this, so coupling predicts a transition from these barrier loci being subjected to only direct selection and acting kind of independently, to a transition to direct and indirect selection effects applying upon several barrier loci. So they will act collectively together as stronger barriers, and they will act more and more as a large aggregate barrier loci. And these aggregate effects can lead to nonlinear transition in LD, ultimately resulting in genome-wide congealing and the speciation at the final stage. So quickly, I want to go over genomic clines because they're kind of central to this work. They represent the gradients of allele frequency change against the hybrid index or the genome-wide admixture. And they are characterized by two parameters. They're characterized by the center, which measure the excess ancestry of an allele. So basically, if the allele is biased towards one parent or another within the hybrid zone. And by the slope, which measure the transition rate of an allele through the hybrid zone. And what's important to note is that steep cline, what you see in red here, do correlate with a stronger selection pressure being applied on this allele. And so one of the expectations of coupling, as I said, based on multi-cline theory, is that as coupling gets stronger, cline will start converging, and the variance along the genome for those clines will be reduced. So that's what you see here in this cartoon. As coupling progresses, you see that the clines start converging and having the same shape and the same parameters. And also, we expect that since coupling is driven by selection, we should see cline getting steeper in the coupled part of the genome. So evidence so far for genomic coupling has remained largely theoretical and simulation-based. There's some great theory and simulation work out there. However, empirical studies have been limited to reduced representation genomic data sets, and that was also in part because there was no method to infer genome-wide genomic clines. So here we conduct the first genome-scale empirical application of this genomic coupling theory. We are using 120 whole genomes of hybrid rattlesnakes, and it was all made possible by our collaborator, Zach Gumpert, so big shout out to him. He developed BGCHM, which allow finally the inference of genome scales genomic clines. And we also developed some new approach to quantify genomic coupling. So quick presentation of our study system. It's a hybrid zone between Crotalus viridis and Crotalus cutulatus in southwestern New Mexico. We can see the map of southwestern United States here in the bottom left. They diverged about 5.5 million years ago. They have ZW sex chromosomes, And in terms of autosomes, they have 7 macro and 10 micro chromosomes. And here in the bottom right, you can also see our structure plot. You see a pretty smooth transition from one parent to another, meaning that our sampling represents the whole gradient of hybrid index that you can find in this hybrid zone. Okay, so how did we measure intrachromosomal coupling? So Barton's coupling coefficient is actually pretty impractical to measure because it's very hard to estimate selection. But as I said before, what we can use is Klein variance. So that's exactly what we did. We estimated for 100 kb non-overlapping windows on the genome, the variance in Klein center and the variance in Klein slope. And then we use both of those variants into a PCA to get a single hyperparameter that will represent our coupling coefficient. And in this case, since it's not exactly the coupling coefficient, we call it the coupling index. Okay, so let's get on to some results now. So first of all, we are going to just check a bit the variance within chromosomes. So it varies, it's already great, but one thing that we can see already is that we have peaks of coupling in the regions surrounding the centromeres. So that's the gray boxes. I hope you can see them. I'm not sure, but the gray boxes around the centromere represents the peak of coupling that overlap the centromere. But we also see some regions that are distant from centromere that show also elevated coupling values. And one thing that I want to mention is that the Z chromosome show overall highly elevated coupling values. Oh yeah, and I forgot to mention, we expect a peak of coupling around centromere because, of course, recombination is greatly reduced in those regions. Okay, and so a very basic prediction of coupling theory is that that coupling will correspond to buildup of linkage in the hybrids. So we wanted to assess this. To do this, we compared the recombination maps of parental species to the recombination map of the hybrids, and that's what you can see in this second panel. So the high pink peaks show region of the genomes where recombination is reduced in hybrids, and therefore where the linkage is stronger. And we can see that the peaks of coupling do correspond with peaks of linkage in the hybrids. And these trends also hold genome-wide. We did some genome-wide regression, and they end up significant and having some effect. Okay, and now going into the assessment of Klein slope. As I said before, coupling should correspond to steep Klein slope since coupling is driven by selection, and that's what you can see in this third panel. So the high red peaks shows region of the genome that have steep slopes, and the low blue peaks show flat slopes. And the peaks of coupling do correspond with peaks in red, so peaks of steep slope. And this also holds genome-wide with a pretty strong effect size. Okay, so now we move on from this within chromosome assessment to actually a whole genome-wide coupling effects. So the theory predicts that coupled traits, because of their multilocus nature, may lead to multilocus interactions that may enhance the coupling genome-wide. So to assess this, we estimated interchromosomal conspecific LD, which is the linkage disequilibrium between variants that are biased towards the same parent. So in this case, variants biased towards Crotalus cucullatus. And so what you can see here in the circles plot, so that's the whole genome. Each section represents a chromosome. The outer track represents the Klein slope, and the bottom track represents the coupling, similar to the panels I've shown before. And in the middle, you can see the interchromosomal LD, conspecific LD for Crotalus cucullatus. So I hope it's not too small. Maybe you'll have to trust me on this. But also what we can see is that we have two hotspots of interchromosomal LD, one on macro 7 and one on macro 6, that also heavily interconnected. And what's interesting is that those two hotspots of interchromosomal LD corresponds to region of the genomes that show among the highest coupling values, intrachromosomal coupling values on autosomes. And we also checked for every windows that is in LD here. We checked for enrichment of Klein slope and of coupling, and yes, we can see that windows that are in LD are enriched for higher coupling values, so intrachromosomal coupling values, and for steeper Klein slopes. So all of this actually show us a little window into how genome-wide congealing may occur and how all this coupling between chromosomes and intrachromosome might work together. But we went a bit further, so we wanted to see if we could assess the biological basis of this. To do this, we used string to predict an interaction between the genes that are in the conspecific LD windows, and we found significant functional enrichment for ion channels and for GABA-gated chloride ion channels as well. So that's the graph that you can see here. That's the network of interactions between the genes, and all the colored dots represent the genes that are ion channel related. So what might this mean? One key phenotype differences between Crotalus viridis and Crotalus cucullatus is actually their venom composition. So Crotalus cucullatus has a PLA2 neurotoxin, crotoxin, that is absent from Crotalus viridis venom. And this toxin is actually known to target ion channels and inhibit also the activity of GABA receptors. So could this be that this is an incompatibility due to self-resistance to their own venom? So we went a bit further into this. We genotyped the PLA2 loci in hybrids and then calculated the LD between these genotypes, so the genotype for this neurotoxin, and the variance in the region surrounding those ion channel genes. And that's the top panels that you can see here. You can see that LD is actually through the roof in the region that contains those ion channels, and also between the region containing those ion channels. And we can also see that the coupling is elevated in both those regions. So all of this shows some evidence that that venom and its biological targets are coupled traits, and that these loci, they manifest elevated intra and intrachromosomal coupling, and that they represent collectively a stronger barrier to introgression. I don't really have time to go into this, but I just wanted to mention that we found some really cool stuff about the large Z effect. So we found extremely elevated intra and interchromosomal coupling. The interchromosomal coupling for the Z chromosome is through the roof. It's about 97% of all interchromosomal coupling that we find. And it's connected to all other chromosomes. So some really cool stuff. And yeah, come talk to me later if you're interested in that, or keep an eye out for our paper. Okay, so in conclusion, we demonstrate here some key prediction of coupling theory in nature, and we highlight the potential of this method for quantitative comparisons of the speciation process across diverse systems. And we also demonstrate some interaction between intra and interchromosomal coupling that might have the role of pulling the whole genomes together towards congealing. And with this, I want to thank my awesome lab, the Castle Lab, and our collaborators, and I'll take any questions.
Super, thanks very much. We have time for one or two questions.
Do you know how old the hybrid zone of these rattlesnakes is? Is this something that's a relatively recent secondary contact, or something that's been going on for a while?
Yeah, we're actually not sure. We didn't time the hybrid zone yet, so I cannot answer. Yeah, sorry.
Some of those blocks you saw of coupling look like there might be structural rearrangements.
Yes, that's definitely one thing that we have to dive into that we haven't done yet. Yes, that's on the plan, is to check for inversions, actually, yes. Yeah. Mm-hmm.
Anything else?
Oh, yeah, sorry.
No, it was very nice talk, really interesting. So you suggest that the venom incompatibility is what perhaps maintains the two species, but aren't there like a lot of viable hybrid rattlesnakes in this area? Like they...
Yes, they are. We expect that's a pattern that we see. So the hybrids will have lower fitness if they don't have the right self-resistance to the venom. We don't know if it immediately kills them, probably not, because we did find hybrids that do not have the right combination, but they probably have a lower fitness, and that's what gives rise to this pattern.
Great, thanks very much.
Thank you very much.
Awesome. Thank you, thank you, thank you. Thank you for organizing the symposium and inviting me here and coming to hear this. It's my great privilege to be able to show some of the awesome work by the incredible scientists in my lab, continuing working on reinforcement in flocks. So I'm going to start with just a broad definition of reinforcement. That is simply selection against hybridization, favoring the evolution of stronger reproductive isolation and sympatry. Pretty broad, and I think our field in general has done an excellent job answering these two questions. Can it happen? Beautiful empirical work across a huge variety of systems documenting reinforcement can happen. And now going on more recently to document the consequences of this reinforcement across many different organisms. Where I think our field is heading is really trying to understand the genetic basis of this process and the genomic consequences. And we've had some beautiful theory and hypotheses formed on what the genetic basis and the genomic consequences should be. But I think what we're lacking are some really clear empirical examples to dive into and validate these ideas. So I'm going to tell you about what we're doing in phlox to further this research. So this is the system we work on, Phlox drumundii. It's a native wildflower in Texas, and it comes in these two beautiful flower color varieties. First, we have this sort of light blue violet color that grows throughout most of central Texas. And then the eastern populations have this deep dark red flower color. And notably, this deep dark red flower color coincides with an area of sympatry with its closely related species Phlox cuspidata. And much of our previous work has very convincingly, I think, documented that this flower color variation is caused by reinforcement. And so through a number of field experiments, we can document strong selection acting on this red flower color in sympatry because it decreases hybridization by 50%. So in sympatry, these dark red flower colors decrease hybridization a lot, generating really strong selection. What we want to know more about now is what is the actual genetic basis of this trait, and can we look now near the genetic mutations causing this flower color variation to inquiry what are the genomic consequences of this kind of selection in sympatry. So when we look at the genetic basis, again, we've been working on this for a while, and we know that there are, in fact, 4 distinct flower colors that actually grow in Texas. This light blue and dark red are joined by a dark blue and a light red here in this area of overlap. And these 4 flower color categories can be explained by 2 loci, each with 2 alleles. So the first locus is what we call the intensity locus, and it corresponds to this R2-R3 MIB transcription factor. And it determines whether these plants make a little bit of pigment in their flowers or a lot of pigment in their flowers. The second locus determines the hue of the flower color. Are you making blue pigments or only red pigments? And it corresponds to this branching enzyme in the anthocyanin pathway, F3'5'H. For the rest of this talk, we're only going to be talking about this R2-R3 MIB transcription factor and our efforts to actually figure out what the mutation is that causes this change in flower color intensity. And this work was done by a phenomenal team in the lab, this PhD student, Austin Garner, a postdoc, Andrea Berardi, and a fabulous technician, Andrew Cameron. So the first thing we did was really characterize how, in fact, this transcription factor might change the amount of pigment made. And so what I'm showing you here is just how we looked at the gene expression throughout developmental time of these flowers to see what's different between the light and the dark. And if we look at the light flower colors, we see expression of this R2-R3 MIB transcription factor peaks right at flower opening to lay down this layer of anthocyanin pigments. Whereas in the dark flowers, we see gene expression is highly upregulated early in development, putting on many layers of anthocyanins before the flower even opens. So some of this work looking at how gene expression and transcription level is sufficient to explain flower color variation leads to the idea that there should be a mutation in the phlox genome somewhere near this R2-R3 MIB transcription factor that causes this flower color variation. And so we wanted to find it. Our strategy is to look at this area where all 4 flower colors coexist and recombine, and we have natural admixture breaking up linkage disequilibrium in order to find an association between some mutation and flower color. Our problem was that it's a needle in a haystack. We have a giant genome, and it seemed rather inefficient to sequence 6 gigabases of a genome of a bunch of individuals to find some SNP near a gene we know it exists at. So instead, we worked with this company, Samplex, to use their X-trop nanopore sequencing, which allowed us to sequence about 80,000 base pairs right around this gene of interest across a bunch of individuals. So shown here is just an example of how we have 100 to 500x coverage of nanopore sequencing in about 50 to 80,000 base pairs right around our gene of interest. So we can target this gene in our giant genome in about 120 individuals that vary in flower color in nature, and then use an association study to try to find what are the causal mutations underlying this. This is our Manhattan plot. It's a little foggy in New York today, right? So what we found was a total of 2 SNPs that are associated with flower color. One of them, what we're calling here SNP1, is perfectly associated, where every light individual is homozygous for this GG allele, and the T allele only exists in dark individuals. So what you can see is this mutation exists right 5' of the start codon. It's about 120 base pairs away from the start codon, right in the middle of the cat and tata boxes. So it's in the essential promoter of this gene. What we did then was start to look at the genetic sequences surrounding this causal mutation and ask how might this be working. And so we can infer transcription factor binding sites, and what we see is that in the light allele, there's nothing really going on right here at this spot. But in the dark allele, we have now introduced the potential for 2 new promoter motifs. One of them is an inverted cat box, and the other is a new binding site for this E2FBF motif. And so we know from other systems that both of these motifs can be associated with upregulation. So this was really exciting. I feel like, wow, we used this strategy to find a mutation that causes a change in flower color and decreases reproductive isolation by 50%. And I think we can conclude that, yes, this huge change in reproductive isolation can, in fact, be due to a single nucleotide polymorphism changing one trait. Now we get to actually look at the genomic consequences, right? I'm an evolutionary biologist. I want to know how this evolved and if we can infer from the patterns of genetic variation across this region, what was the evolutionary history of this mutation. And so again, we go back to some ideas and hypotheses that have been posed in the literature that actually reinforcement and character displacement gives us this great opportunity to compare where a selective mutation has risen to high frequency versus allopatric populations that maintain the ancestral variation and mutation and alleles. And so we might expect that in these allopatric populations, we see that genetic variation shows dancing around neutrality, right? But in sympatry, where these novel mutations have swept, hopefully due to selection, we would see some sort of deviation from neutrality, right? It's a beautiful little contrast here. So here, some summary stats to GMSD, Phe and Wu-H, you pick whatever you want out of your hat, and hopefully you'll see a signature. So since we had these, 50 to 80,000 base pairs surrounding this causal mutation, we started doing some sliding windows to see what does this variation look like. Not much. Okay, so this is to GMSD, and we can see from allopatric and sympatric populations that, we're bouncing right around what we might expect with neutrality. And there's nothing different in the genetic variation from allopatry and sympatry. Same is true for Phe and Wu-H, same is true for pi, same is true for haplotype structure. It all looks like this. Same is even true when we do FST scans comparing Drummondii to that closely related species Cuspidata. Nothing stands out. Okay, so there's no signature of a selective sweep around this mutation that we know we've measured selection on in sympatry, does not exist in allopatry. Why? Why is it that this theory and these ideas and hypotheses that have been so established in the literature are not panning out in my mutation, right? Okay, so there's a lot of reasons that also the literature has proposed. Could it be something about these demographic parameters of our species, effective population size, patterns of migration between allopatry and sympatry, time since mutation? Maybe the initial frequency of this mutation when selection started acting on it in sympatry. So all of these ideas are bubbling up, and what do we do about it? Our strategy was to actually now infer some of these demographic parameters from flocks and try to simulate what would a selective event look like in sympatry, and then start to play around with these parameters and see at what point is this signature that we're looking for sensitive to what we're seeing, to these parameters. So this is work done by a fabulous postdoc in the lab, Patrick, who used this diffusion approximation for demographic inference, DOTI program, using a whole bunch of neutral genetic variation across allopatric and sympatric populations to infer some of these sort of fundamental demographic parameters. I just want to point out, any of 1 million? Man, wildflowers are awesome. Okay, so we have these giant effective population sizes. We have reasonable migration between allopatry and sympatry. We've got what looks like a fairly old split, maybe 300,000 generations between these sort of the heart of the allopatric and sympatric zone. So we can now use SLIM, or the simulation framework, to try to put a selective mutation in sympatry in a flocks population and see what happens, right? So we plug these parameters in, we run SLIM, whew, we see a signature of selection. So here's one example of an output of SLIM where we can then look at the genetic variation, scan across it, see indeed there is a dip into GMUS D, just like we would expect there to be in a population where selection is acting on a new mutation. We do this, 100 more times. I'm showing you just 5. And sure enough, it's pretty reliable that you're going to see a signature of selection. Okay, so under flocks parameters, we can see that there's a signature of selection. But in our data, we don't see that there's a signature of selection. So what Patrick then did was start playing around with some of these, what we might call logical parameters, right? Some of them we may have just inferred incorrectly in our DOTI simulation, so we can play around with them. And some of them, like initial frequency, we have no idea. We can't go back in time and know. So maybe it's just a classic soft sweep. So what he did then was take, one of these parameters and he changes it. Like, what if flocks does not actually have an NE of 1 million? And it's something slightly more reasonable, like 300,000. Guess what? It goes away. Okay, so we lose that, and we can also do it with something small, like 50,000 NE, and sure enough, the signature goes away. And so he's been playing around with all these different parameter varieties, and I'm just going to summarize this by saying there's actually a pretty small parameter space under which we are seeing a signature of selection. And this really got us thinking that maybe, actually, the mutations that we care about in the phenotypes that we care about are not visible in our current tests and ideas of what is, the patterns of genetic variation that we're seeing from these causal mutations. And so I don't have an answer for you. I can't go back in time and tell you why, in our particular case, we don't see a signature of selection, but it could be because our demographic parameters were wrong, or it could be because this is hanging out in the flocks population for a while before it was under selection. But what I can say is that maybe there's a lot of signatures that we can't see of selection because of the way evolution works and drift. And so in conclusion, I think that we have, as scientists and in this field, done a beautiful job documenting patterns of natural variation in order to understand these processes that we're really interested in, and done incredible experimental work to dig in and document these processes of evolution involved in speciation. And I think where I hope to see the field go is a deeper understanding of these mechanisms underlying some of this phenotypic variation, so then we can link now with empirical data how these mechanisms are then leaving patterns in genomic data. And so with that, I thank the fabulous group I have a privilege of working with every day, particularly Austin, Patrick, Andrea, Felix, and Andrew, my funding sources. And I draw your attention to a few more talks from the group if you want to see more flocks this afternoon, so stick with it. Thank you very much.
Is there a very quick question before coffee?
I was wondering, is it possible to quantify the changes in LD away from the mutation to
I'm glad you asked that. Yes, and we have, and I had to take that out for size, but it drops off very quickly. We see that within a few hundred base pairs, there's nothing. Good question. Yeah. Yeah. Oh, the question was, what does LD look like around the causal mutation? Not much. Yeah.
If the signature is rare, what does this mean for systems where we can't get to like the causal gene?
I don't
See the signature,
know. I don't know, right? Like, I'm not saying that those signatures aren't underlying something interesting, right? There's probably something there, but what else are we missing? I don't know. It's everywhere. I don't know. Thank you.
Welcome back to the symposium. Next up, we have Alexandre Blanckaert talking about interactions between mechanisms of reproductive isolation.
Okay, so hello everyone, and thank you first for the organizer for giving me the chance to present my work here. So as we have already heard today, so speciation can come, and reproductive isolation can come in many forms, and usually we try to characterize it depending on the timing of when it happens, whether it happens before or after the formation of the zygotes, and also the nature of the selection underlying this mechanism, whether it is imposed by the environment or whether it is due to the genomic buildup itself that there is something going on. And obviously, for the rest of the talk, I pick one example of each of these mechanisms, and I will be focusing on it. So matches for intrinsic postzygotic isolation, genetic incompatibility for postzygotic intrinsic isolation, and local adaptation both for prezygotic extrinsic, in that case it's selection against parents, or for postzygotic extrinsic, in that case it's selection against F1 and other F generations. And obviously each of these mechanisms on their own have received a lot of attention, but not only each of them, but their interaction, for example, as we have seen already earlier today, like the interaction between postzygotic isolation and the evolution of matches, which is coin reinforcement, has received lots of attention, but also the interaction between local adaptation and mate choice, which correspond to all of the magical trait models that I think we have heard about as well. And finally, we have seen, it has been shown that when there is migrations, when there is migration, DMIs will not be able to accumulate without at least local adaptation, which we have seen in the talk this morning as well. So my idea here is to just try to have all of these mechanisms together, but not only like this, but also to have a genetic basis that is common to see how they are going to interact and evolve together. So just to quickly summarize the main questions, in a scenario of speciation with migration, how each of these mechanisms for reproductive isolation evolve and how they are going to interact. So to do this, we are going to consider the following model, where we have two populations that are going to exchange migrants, and we have our three mechanisms of interest. And to show you how they are interdependent of each other, I'm going to illustrate first a brief overview of how they work, and then we will go a bit more into details. So we have a genome with many loci, and all of these loci are going to contribute additively to the phenotype, and this phenotype is going to be under selection, and therefore that's how local adaptation works. But this phenotype will also be used as a mating cue for individual, because we are going to use a phenotype matching preferences system, and therefore the phenotype is also going to be interacting with mate choice. And finally, the exact same loci that are used to encode the phenotype are going for some of them to form their own incompatibilities that are intrinsic. So simply having these two together is bad regardless of the phenotype that is resulting. So that's how we have this interdependency, and now a bit more detail about how each of these mechanisms are modeled. So for the local adaptation, we are going to use Fisher geometric model, where we have these many loci that contribute additively to the phenotype, and then we have a simple quadratic relationship that is illustrated here between the phenotype and the fitness. So for each environment, you have an optimum, and then you have this Gaussian shape, but it doesn't have to be Gaussian decay of the fitness, that when you have the optimum, you have the best fitness, and you go away from the optimum in the phenotypic space, you have a worse fitness. For the mate choice, as I mentioned earlier, we are going to have a preference by phenotype matching. So in that case, we model male mate choice because this is very vaguely inspired by spider mites. And so the idea is that a male, in that case, when he mates with a female, he can compare his phenotype to the female phenotype, and depending on this distance in the phenotypic space, he will either accept or reject this this mating. And then we also model it by a Gaussian to represent a bit the noise that can be in natures. And the only parameter that matters here is if you pick a perfectly adapted individual from environment 1, and he has to pick from an individual from a perfectly adapted to environment 2, this is what this parameter corresponds to. And if you have p equal 1, there is no choice. And you can see as it gets pickier and pickier, only individual really similar to the male will be accepted. And finally, for the intrinsic postzygotic isolation, we are going to model simple Betson-Dodson-Schmuller incompatibilities. In that case, we have 50 pairs. So what's in the middle I have represented is the genome with these 5 linkage blocks that can be chromosome, not chromosome, depending on their distance. And you have these incompatible pairs that can happen within linkage block or between linkage block. And all of these barriers are going to act together and may impact the genetic barrier that may exist between the two populations, and we are going to track them and measure this barrier by simply looking at the sojourn time of a neutral marker when we switch population. More precisely, we are considering this case. Once our population have evolved for a long time, you pick one individual from environment 1. Now that they're isolated, you put it in population 2, and just have this neutral marker that is independent, that comes with it, and you can track what happened to this marker. And you do this many times, and you can look simply if this marker is often lost or not. And if you expect a weak barrier, then you should have this distribution with a long tail that the marker can sometimes remain for a long time in the population. And if the barrier is extremely strong, you should expect this marker to only stay for a couple of generations before being always extinct. So now I'm going to present you some results first, and what is happening when we have strong migration rate, which is up to 20% every generation between the two populations. And this is, we are doing the simulation with all the barriers that can evolve together. They all start from ancestral state, they are equivalent, but all the barriers can evolve together, but I'm going to show you the outcome one barrier at a time because it's a bit easier to visualize. So first, local adaptation. So now we have this plot, and they are going to be a bit similar each plot for the next few slides. So on the x-axis, it's the time in n generation. So we let the population evolve for up to 50 n generations. On the y-axis, you have the metric of interest. In that case, it's the phenotype, the mean phenotype of the population. And then you can see for 0, it corresponds to the ancestral phenotype. Up to 5, you have the optimum of the environment. The dashed line is what you should expect if you had infinitely strong selection because of migration selection balance. And the color lines are each of the replicate, and the black line is the mean across this replicate. And what we can clearly see is we evolved some local adaptation, but it seems to be not that strong. But this is, we can look at what's happened in the last generation and look at the population itself to get a better snapshot of what's going on, and this is what we obtain. So each color now is a replicate again. The x-axis is the phenotypic values that all the individual can take, and this is simply a distribution plot. And what we can see, we have like four remarkable peaks, ones that correspond to individual almost perfectly adapted to the environment in question, which is environment 2. We have a smaller peak much further to the right, which correspond to basically migrant individual or offspring of migrant individuals. And then you have F1 individual and F2 backcrosses as well. So clearly, despite this really strong migration, we have clearly local adaptation working on quite quickly. So now we can move on to mate choice, which it worked. So now this is the same plot than before, except the metric on the Y is a preference. And you can, on the top, it means no preference. On the bottom, it means really strong preferences. It's a log scale. And what you can see first is that in all cases, we evolve pretty strong preferences, like a male from population 1 will pick a female perfectly adapted from population 2 in less than 1 in 100 cases. And we can see there's a bit of delay. And if you compare it to the small inset on the top left, you can see it's basically, it waits until all localization has done all the work before mate choice can evolve, because they're using kind of the same queue, meaning that localization is always, in that case, going to precede mate choice in its evolution. Finally, we can look at the evolution of DMIs and whether they will accumulate or not on the genome. And so now it's always the same plot, except this time the y-axis is simply how much fitness is reduced to the presence of DMI in the genomes. And what you can see is on the bottom, it means no DMI accumulates, and on the top, it means that some DMI have accumulated. And in almost all cases, nothing happened. There was no DMI accumulation. And you can see from the inset on the left, we are in a situation where we have strong local adaptation, we have strong mate choice, and yet nothing happened. And this is something that was already pointed out by one of the previous speakers, that despite this condition, DMI still struggle a lot to accumulate. So what does it all mean for the sojourn time and the genetic barrier? So now we are looking at slightly different figures. So now that our population have evolved, we can turn off some of the genetic barrier, and in that case, for the first display, only keep local adaptation and compare the sojourn time, the mean sojourn time of a mutation to the neutral case, which is represented by the dashed line. And you can see that local adaptation is doing a really good job at reducing this sojourn time, meaning that there is a strong barrier to gene flow. Mate choice is doing quite okay-ish with a bit of a difference between males and females because males is the one that is choosing. And DMI is pretty pointless, but also they don't evolve, so it's not really surprising. We can also combine this barrier, in that case, local adaptation and mate choice together, and do the same, redo the same measure. And we can see it reduce a bit more the sojourn time of the mutation, but not as much as we could have expected based on the independent effect of the barrier, suggesting that it's not antagonistic, but there is a bit of a diminishing return. Obviously, we can fill up the missing part, and we can see that mostly three main take-home messages out of this. Local adaptation does most of the job, and on its own. When you combine it with other barriers, it's not antagonistic, but there is clearly diminishing return. And I'm forgetting one. Sorry. And yes. And integration, you can see that the difference between male and females, the males have a bit of a less reduction in general, so integration should be favored through the sex that is doing the choosing when there is mate choice. Obviously, this was with like really strong migration, and we can have a brief peek at what's happening when we vary migrations. So now you can see for m equals 0.2, sorry, on the bottom left, this is the plot you have seen before, and now we are reducing migration across each facet. And mate choice, we can only evolve when there is strong migration. When there is really weak migration, it will not evolve, and meaning that migration is promoting in that case the evolution of mate choice. We can do the same for DMIs. Is that the, yes, it went. And so now it's kind of the opposite patterns. Again, you have seen the plot for m equal 0.2 before, and now as we reduce migration, we can see only when migration is null or really, really weak, then we can have DMI accumulations. I'm not showing local adaptation because we have seen it works for 0.2, so obviously it works for anything below. For the sojourn time, I'm simplifying it, and I'm only comparing m equals 0 to m equal 0.2 that we have seen before, and there is basically almost no differences. Now that DMIs can accumulate, for m equals 0, we can see it does reduce a tiny bit the sojourn time, but not that much, and most of the work is still done by local adaptation. So to conclude, we have seen that first, local adaptation is always the first barrier that is going to evolve first, and it's going to be followed by mate choice or the accumulation of DMIs, but they tend to be exclusive. Second, we have seen that migration does not really affect local adaptation, and it will promote mate choice evolution and hinder the accumulation of DMI. We have seen the third one is like the different barriers act together, but there is a bit of a diminishing return. And finally, we have seen the main thing that seems to matter a lot here is local adaptation driving the system. And in all of this result, it kind of raises questions like, are the DMI really actors in the speciation process, especially when there is migration, or are they a simple testimony of the speciation progress having happened? With this, I would like to thank my supervisor, Vitor Souza, collaborators in Fragata, Vitor's group, the funding agency, and you for your attention.
Thank you very much. There's time for some questions.
I may have missed it, but could you say what the strength of local adaptation selection was there?
So because we are using this, it was kind of hidden. Because we are using this Fisher landscape thing, so it's roughly, there is not a single value, but so the perfectly adapted alternative phenotype has a 10% fineness. But obviously the algorithm have intermediate finesse and everything because we are using all of these correlation functions. So obviously selection is stronger than migration in the system, otherwise it will not work at all. But it's not it's not step functions, so it's a bit, does that make sense?
And it's not as strong as some of your mate choice parameters, possibly.
Well, it's the same for mate choice. Again, because we're using this Gaussian, the value I gave corresponds to this perfectly, the choice between two perfectly adapted individuals. But like already, an hybrid individual usually have like more than 2/3 the value of the bad parents. So it's really this kind of continuous things about all of these mechanisms that create these patterns emerging.
Time for one quick question.
Yeah.
Go ahead.
So your mate choice was matching. I was wondering if you have some ideas about adding like a signal and trait kind of mate choice
I mean, so you would have to want a more step-like function instead of this Gaussian kind of thing, so that the signal is much clearer. So it should help, because obviously if everything intermediate, in particular the F1 hybrids, they will not suffer that much. And technically they are doing, if you do the average between two parameter hybrids, there is always a bit of heterosis because of all the curvature of the landscapes. So the hybrids are doing better than you would expect, so that kind of promotes gene flow between populations. So what you suggest, I think it will add to invert gene flow.
So let's stop there and move on to the next speaker. So next up we have Katharine Khoury, disentangling pollination efficacy and its role in maintaining subspecies boundaries in an African storksbill.
Okay. Good morning, everybody. My name is Katharine. I'm a master's student from South Africa, and I'm going to be chatting to you today about some research I've been doing into pollination efficiency and its role in maintaining the subspecies boundaries between these two Pelargonium species. So reproductive isolation is when we quantify gene flow barriers in order to understand what maintains species boundaries. In plants, these are broadly grouped into pre- and post-pollination barriers, with pre-pollination being all the barriers that work before pollen lands on the stigma, and post-pollination working after that. And these work sequentially from pre to post-pollination. Today, I'm largely going to be focusing on a single barrier, floral isolation. So floral isolation has largely been studied in three ways. The first way is by, in geographically isolated populations, it's usually measured by looking at the overlap in pollinator assemblages, whereas in sympatric populations, it's usually quantified by measuring behavioral or mechanical isolation, with behavioral isolation being when pollinators make preferred visits to one flower over another, and mechanical isolation being when different flower architectures prevent interspecific pollen transfer. Over here, I have extracted some data from a recent Christie review that summarized the strength of reproductive isolation barriers in seed plants over the last 15 years. On the left-hand side, I have the number of taxa pairs and then all the different ways that pollinator isolation has been quantified. As you can see in the graph, pollinator isolation has been quantified a lot in sympatric populations. And there's actually only one study each in parapatric and sympatric populations that have actually studied both behavioral and mechanical isolation. This gap in the literature was also identified by another review from Kay and Sargent, and this is the summary points right from the end of their paper. They point out that we need to relax the assumption that floral isolation must arise in sympatry in order to play a role in plant speciation, so we need to be studying it in geographically isolated populations. They also point out that we need to be including measures of pollinator importance, which is just the combination of that behavioral and mechanical isolation. I'm going to go into a little bit more detail of why, whenever we study floral isolation, we need to be including both of these barriers. So we can get very strong floral isolation through a few ways. The first is if we have strong behavioral isolation where pollinators make a lot of preferred visits to one taxa and not the other. In this case, because they never actually make visits to the heterospecific taxa, the role of mechanical isolation is negated. In the second scenario, you might have a pollinator that makes frequent transitions between closely related taxa, but because of differences in the flower architectures, we might find that there's still no heterospecific pollen transfer, resulting in strong floral isolation. In this case, if we had only measured behavioral isolation, we would have missed the overall strength of floral isolation. The third scenario is where we measure both behavioral and mechanical isolation using pollinator efficiency data. And this is usually the way that it is likely acting in the field. Floral isolation can also interact with other reproductive isolation barriers, such as immigrant and viability. So over here, I have two geographically isolated populations that are adapted to two different sets of functional pollinator groups. We may have some kind of a dispersal event which can be simulated using something like a reciprocal translocation. And if, because the native phenotype is adapted to the local pollinator and the introduced phenotype is not, you may find that the introduced phenotype has a lower fitness because of selection against them through pollinator behavior and morphological fit with those flowers. So it's about time that I actually introduced you to the study species I've been working on. These are the two subspecies of Pelargonium dipetalum. On the left, we have subspecies dipetalum, which has these very short tubes and pink flowers, and on the right, we have subspecies stenosyphon, which has these very, very long tubes and white flowers. From doing pollinator observations across 3 sites, we found that subspecies dipetalum is largely pollinated by short proboscis flies and small solitary bees. For the other subspecies, we found that they are exclusively pollinated by a singular species of long proboscis fly. Because of the variation in morphology between these species and the pollinator communities, we then asked a series of research questions. In order to ask this, we've also placed these two subspecies in artificial sympatry. So we measured the strength of behavioral and mechanical isolation, and then we combined those together in order to get a measure of pollination efficiency. We then also asked if there was immigrant and viability at the flowering stage. We also measured the strength of post-pollination isolation, and then we asked what the contribution of each of these barriers is to total isolation. So this is just a map of the distribution. As you can see, they occur in the Cape Floristic region of South Africa in the southwest. Subspecies dipetalum, which is in green over here, has a much wider distribution than subspecies stenosyphon, with those stenosyphon populations being largely encased within the wider dipetalum population. In those triangles, you can see our reciprocal translocation sites. So these are the sets of results from behavioral isolation. In the gray here, you can see the results from the functional pollinators that were present at the one site, and in the white, that's the functional pollinators that were present at the other site. We found that solitary bees had a much stronger first preference for their native dipetalums over the introduced stenosyphons, but the short proboscis flies did not show this trend. We also find that long proboscis flies also had a higher probability of first preference for their native stenosyphon over the introduced dipetalum. For mechanical isolation, when we looked at pollen grains deposited per visit, we found that solitary bees and short proboscis flies did not deposit significantly more pollen on their native dipetalum compared to the introduced stenosyphon. However, for those long proboscis flies, we found that they deposited much more pollen grains per visit on their native stenosyphon compared to the introduced dipetalum. In addition to measuring pollen deposition, we also recorded whether or not pollinators made contact with the reproductive parts of these flowers, and we found very, very similar results to what you can see here. When we finally look at pollinator efficiency, on the left-hand side, we have the visitation rates, and you can see that all 3 sets of functional pollinators here have a higher visitation rate to their native phenotype over the introduced phenotype. But this is much more exaggerated when we actually look at pollinator efficiency, which is the combination of that behavioral and mechanical isolation. So long proboscis flies are by far the most efficient pollinators to their native stenosyphon, and for dipetalum, the solitary bees and short proboscis flies are also by far the most efficient pollinators for them. This is the seed set results from our reciprocal translocation. So over here we can see that the native phenotypes at each site have a higher seed set compared to the introduced phenotypes. And for post-pollination isolation, we also found that conspecific treatments for both subspecies had higher seed set compared to the heterospecific treatments. For the contribution of each of these barriers to total isolation, well, we then looked at the contribution of each of these barriers to total isolation. Over here, you can also see that we've included ecogeographic isolation. Because these are still largely geographically isolated populations, we included this to also look at that contribution in maintaining reproductive isolation. Because the range of the stenosiphons is largely encased within the range of a much wider range of the dipetalums, we actually found that there was very strong ecogeographic isolation in the one gene flow direction, but very weak ecogeographic isolation in the other gene flow direction. For the dipetalums, we also found that there was relatively strong behavioral isolation, but weaker mechanical isolation. But in the opposite gene flow direction, we found that there was relatively weak behavioral isolation, but very strong mechanical isolation. We also found that there was strong immigrant and viability and post-pollination isolation in both gene flow directions. For the overall contribution to reproductive isolation, in the one direction, we found that ecogeographic isolation made the largest contribution, but in the other gene flow direction, we found that it was the combination of behavioral and mechanical isolation that is maintaining reproductive isolation between these subspecies. So overall, we found very strong evidence of floral isolation between these Pelargonium subspecies that is mediated by both pollen behavior and mechanical fit. We also found that both floral isolation and post-pollination isolation cause immigrant and viability between these Pelargonium subspecies. And we also found that ecogeographic, floral, and to an extent post-pollination isolation maintains strong reproductive isolation between these subspecies. I'd just like to say thank you to all of our funders and the people that have assisted with the research. I'm happy to take any questions.
Thank you very much. There's time for questions. Yeah, go ahead.
Thank you for your talk, Katharine. Very nice. They seem to be quite reproductively isolated, right?
Yeah.
What do you say? Would you say it's almost different species then, or?
Yeah, so these two subspecies were actually initially described a few years ago, and they were then classified as different subspecies. Realistically now, I think it would be reasonable to say that they should actually just be considered different species because there's such strong pre- and post-pollination reproductive isolation.
Can you say anything about, in terms of a biogeographic scenario, explaining this small pocket of one subspecies?
Yeah, sure. I actually think that's probably mostly due to the distribution of that very specialist pollinator. So those long-tongue flies are only found in very specific areas of South Africa, and it's quite a specific relationship of that's only that one species. And there's a pollinator guild for that species that occurs specifically in that area. Obviously, it's quite difficult to really test this properly, but my guess is what happened here is that you had this very wide geographic range for that initial that short tube subspecies that then made a pollinator shift to that long-tongue fly. And then over time, you also had that very strong immigrant and viability and post-pollination isolation developing.
There would be time for, yeah, go ahead.
So I was interested in the results about for the short tube species, its two pollinators. The bees, if I remember correctly, the bees know what's up, they don't waste their time, and the flies don't.
Yeah.
Do you have any ideas about why they are so
Different.
why they waste their time?
Flies in general, even with the long-tongue flies, they don't seem to have, although they do still have some preference for their native phenotypes, I found overall that flies don't seem to show a strong behavioral isolation compared to bees, which in South Africa seem to be quite picky over the flowers that they visit. I'm actually not sure what it's caused by, but I think it's it could be due to all sorts of things, differences in visual systems or particular cues with the flowers or whatever it is. Yeah.
Thank you. I think now is a good time to move on to the next speaker.
Researcher in the Biological Clocks Group at the Max Planck for Evolutionary Biology. And today I'm going to talk to you about magic traits. So in 1943, when Ernst Mayr defined biological species as groups of interbreeding natural populations that are reproductively isolated from each other, he underscored the central role that reproduction plays in what makes a species a species. Understanding speciation is therefore largely a matter of understanding the reproductive barriers that exist between diverging populations. And through time, it has become apparent that the class of barriers known as magic traits are those that are most favorable or most likely to result in speciation. So a magic trait is a trait that plays a role in both the ecology and the mating of an organism. More specifically, it is an allele or set of alleles under ecological divergent selection, which through pleiotropy result in one or more barriers to reproduction. Classically, mate signaling traits have been used to look for evidence of magic traits, with one of the most famous examples being in the heliconius butterflies, where the wing color plays a role in Mullerian mimicry, while also serving to signal to potential mates that this is a member of the same species. However, similar examples have been found in poison dart frogs, or the African weakly electric fish, which use electrical pulses to find prey and also find potential mates, or in these coral reef hermits, which use coloration to find mates, but also use it for camouflage. However, one of the main challenges in determining if a trait is a magic trait is determining that the trait is actually the result of pleiotropy rather than the pleiotropy or sort of effect of pleiotropy being replicated by a complex of genes in tight linkage. However, a class of magic traits which sort of sidesteps this issue are known as the automatic magic traits. And these traits assort mating in time and space and thus automatically result in reproductive isolation. Examples include habitat preference, like for the Ragoletis fly, which can either mate on apples or on hawthorn berries, or timed reproduction, where populations reproduce at different times of the year and thus miss each other. And these automatic magic traits have been suggested to be the traits most likely to result in speciation. Furthermore, at least for timed reproduction, it's found across the tree of life. This is just a little phylogeny showing all of the known lunar spawning rhythms. However, you can also think about flowering times in plants or insects mating at different times of the year. So given the likelihood of these traits to result in speciation, as well as their apparent commonality, the automatic magic traits might actually be very important diversity-generating mechanisms. However, to assess their importance, it's first important to ask, what is the efficiency of a given trait? Of course, if a magic trait exists, but it isn't very good at reducing gene flow between divergent populations, then it's probably not very important for generating diversity. And studies have shown that very rarely does one trait actually result in reproductive isolation. Rather, it's the coupling of multiple traits over the speciation process that is required to sort of complete the speciation process. And certain genomic architectures are a little bit more amenable to coupling. For example, if a trait under divergent selection is more polygenic, then there are more genes under divergent selection in the genome, and there's a higher chance that one of these genes is going to be close to a barrier locus and thus can sort of catch the barrier locus, making coupling more likely. So to recap, magic traits link ecology and reproduction and promote speciation, with the automatic magic traits being suggested to be the most favorable for speciation. And if these are important, then the barriers should be efficient and also have an architecture that's amenable to coupling. And so an automatic magic trait with a sort of favorable genomic architecture could be highly important for driving the speciation process. So I'd like to give you a little example of an automatic magic trait in the species I study, Clunio marinus. And we study these guys because they time their reproduction to the phases of the moon using an endogenous biological clock. So Clunio spends the majority of its life as a little worm, a little larva, living in these algal turfs at the bottom of the intertidal zone. And these turfs are only exposed to the air during the lowest of the low tides, which happens around new moon and full moon. So during these low tides, the individuals will emerge from the water, and the adults will scan around on the water surface with the males looking for females. And once the male has found a female, he will take her to these little algal turfs that you can see just at the edge of the water, where he'll drop the female off, and the female will lay her eggs, and then the adults will die, and then the larvae will hatch and live their lives waiting for the next reproductive cycle. And so you can imagine how given that the females sort of require these exposed substrates to lay their egg, there would be strong environmental pressure on the timing of mating, strong selection on the timing of mating. And thus, if there was some sort of divergent selection, this could easily turn into an automatic magic trait. And indeed, in 2 places on the coast of France, on the tip of Brittany Peninsula, we find multiple sympatric chronotypes. So these are populations that have diverged in the timing of reproduction, but actually live right on top of each other. And so a genetic study found that these different chronotypes were the product of a single recent colonization event, but also found that there was little genetic differentiation between them. It would suggest either that the timing isn't a very efficient barrier to gene flow, or that the chronotypes diverged very recently. So this leaves us with 3 questions. 1, how important or efficient is Clunio's reproductive timing? 2, what other barriers to reproduction might exist if the timing isn't that efficient, if it's a little bit leaky? And 3, what loci might underlie these barriers? So to answer question 1, we went out to the field in Roscoff where we have 2 chronotypes, one that emerges on the full moon and one that emerges on the new moon. And over the course of 2 lunar months, we sampled copula. So these are individuals that are actively mating with each other. And we took these individuals back to the lab and designed a multi-locus genotyping assay based on high FST peaks, so regions in the genome that have diverged between these 2 chronotypes. And by genotyping these high FST SNPs, basically, in these regions, we can assign individuals basically into full moon chronotype or new moon chronotype. And our expectation is that if the timing is an effective barrier to reproduction, then the genotype should correspond with the time of the month, as well as the couple genotype, so within a couple, should match each other. So full moon should be mating with full moon, and new moon should be mating with new moon. And when we look at the data, with the x-axis here being the day that we caught them in the lunar month, and the y-axis being the time of capture, we see that in general, the genotypes do correspond with the time of the month. So the yellow individuals are emerging during the full moon, and the blue individuals, which are genotyped as new moon, are emerging during the new moon. But what you might notice is that there are a lot of intermediate color individuals here, sort of hybrids. And so especially towards the end of the peaks, it seems like there's a lot of hybridization happening. So to further quantify the gene flow that's taking place, we classified individuals based on their genotype into either full moon or new moon. So if their genotype was greater than 0.5, they're considered full moon. If it was less than 0.5, they're considered new moon. And we found that about 6% of the individuals that we caught emerged opposite to their expected genotype. So these are individuals that were genotypically new moon that would come out during full moon or vice versa. And then to look at this on the couple level, we found that about 8% of couples were actually of opposite genotype to each other. So to come back to our questions, we find that Clunio's reproductive timing is an important barrier. It does successfully separate the reproduction between the 2 chronotypes, though it's about 8% leaky. So what other barriers to reproduction might exist to sort of supplant this leaky barrier? So to look for other barriers, we did a series of laboratory crosses where we crossed full moon to new moon individuals in the lab to check if there are any barriers along the sort of life cycle that you can see on the screen here. And the first place we checked was if there's a barrier that outside of timing that prevents the males from mating with the females. And the way we did this is we took males, one male and one female, and we put them in this little arena that you can see, and we timed how long it took for the male to copulate with the female. And the expectation was that if there was some sort of assortative mating, then the between chronotype crosses, so like full moon to new moon or new moon to full moon, would take longer than the within chronotype crosses because there would be some sort of reluctance of the male to mate with the female. And when we look at the data, that's exactly what we see. So here we use Cox proportional hazards regression, and we found that crossing type was a significant predictor of how long it would take to cross, how long mating would take. And interestingly, the crossing type and female age interaction was also very significant. And the female age is how long the female had sort of sat around after she had emerged before we added her to the arena. And you can see this in the graph here, where the within chronotype crosses, there's not a huge difference in how long the cross would take between the young females and the old females. But for the between chronotype crosses, when the female was old, the cross would take significantly longer, or not even happen at all. So this is evidence that there is an additional barrier preventing males and females from different chronotypes from mating with each other. But what if they do mate with each other? Maybe there's some barrier that prevents fertilization. So to do this, we let those individuals that we crossed in the lab lay their eggs, and then we came back a few days later and we counted how many eggs were fertilized. And if there was some sort of gametic incompatibility, then we would expect the fertilization rate for the between chronotype crosses to be lower. But we did not find any evidence of that. So we can rule out the existence of a barrier at this step. Okay, so what about in the next step? Maybe there's some sort of hybrid depression where the hybrid larvae don't survive or don't hatch as well. And so to check for this, we came back a few days later after counting the fertilized eggs, and we let the eggs hatch into larvae, and we counted how many larvae had survived hatching. And our expectation is that the hatching rate for the between chronotype crosses would be lower if there was some sort of hybrid depression. But again, we don't find any evidence of that. In fact, we find the opposite trend to be the case. So we can also rule out that there is a barrier to reproduction here. And I'll come back to that last question mark in a few minutes. So going back to our questions, what other barriers to reproduction might exist? Well, we found assortative mating. So what loci might underlie these barriers? So to do this, we're going to go back to the field couples, and just thinking about what we actually genotyped to get our genotype assay working was we went after these highly diverged regions. And these regions are diverged in theory because they are resistant to gene flow, which would suggest that there is some sort of reproductive barrier locus there. So from the perspective that these are barrier loci, then we can sort of ask the question, okay, then which loci are associated with which barrier? And we did this using multiple regression and skipping over a lot of the statistics here. Basically what we found for our different barriers, so the circulunar timing would be the day of the month that they emerge, the circadian timing is the time of the day that they emerge, and the assortative mating is what we saw in the lab. We find several loci associated with each. Then for the gametic incompatibility and the hybrid depression, like in the lab, we didn't see anything. Again, in the field couples, we don't see anything. But the thing we couldn't test for in the lab was hybrid sterility because we didn't have any hybrid individuals. But in the field, as we saw in the graph in the beginning, we did catch a number of hybrid individuals. And so when we associated that with the number of eggs a female would produce, so the genotype by the number of eggs that she would produce, we found that there are actually two loci which were significantly associated with hybrid sterility, where the female, if she was heterozygous, would produce significantly fewer eggs. So if we look at which loci are associated with which barriers, there are sort of two observations that we can make here. One observation is that for the barriers that were associated with some sort of, for the loci that were associated with some sort of barrier, most of them were actually associated with more than one, maybe even three barriers. And so it seems like there's actually extensive pleiotropy taking place between these reproductive barriers that result a single locus resulting in more than one reproductive barrier. And the second observation that you might make is that every single loci that is associated with a barrier is associated with a timing barrier. And so this kind of leads us to the sort of scenario, the following scenario, which is that divergent selection sort of acted on Clunio's clock and drove divergence in the clock, which created this automatic assortment that we would expect from the automatic magic trait. However, this divergence in the clock seemed to pleiotropically also create mate choice and hybrid sterility. So it created more barriers through pleiotropy. Right, so if we go back to our final question, what loci might underlie these barriers, we find that the clock loci and the other barrier loci are actually one and the same. And taking this all together, we can conclude that Clunio's reproductive timing was actually very important in driving the incipient speciation event that we observed. So to put this into a more general context, we knew from the outset that Clunio's clock was omni or polygenic, and we know that these polygenic traits are more likely to catch barriers. And if we think about clocks more generally, clocks are sort of known to be polygenic. It's part of their nature. They're these molecular systems where you have multiple interacting genes, and so they're sort of polygenic systems. And we also know that timed reproduction is actually very common across the tree of life. And if some sort of divergent selection were to act on this timed reproduction, then it would make it a magic trait. And so this kind of leaves me with one last question, which is how often have clocks driven speciation events? And I think if we consider what we've learned from Clunia marinus, the answer to that question might be more often than we would expect. So thank you very much for listening, and a huge thank you to my group and Dr. Runa Ekram for helping with this project. And if you have any questions, please email me at the email address down below. Thanks.
So I want to start introducing the people that have actually done the work that I will show here. I don't really see much of the screen, which are Mariana, Carlos, Seraina, and Ana, and Pilar that is with us today here. So I want to start saying that natural selection is the major force driving diversification. And there are many examples that show that. But of course, not everybody, not all data agree with that, and not everybody agrees with that. And this recent meta-analysis published about a year and a half ago, that probably you all know, with empirical data, so that not all cases conform to this scenario. However, there are many examples, particularly in adaptive radiations, where organisms adapt to different habitats, and there's diversification and speciation, and I brought here a classical example with anoles. And there are also plenty of examples of organisms adapting to different food resources, and this is a classic example with cichlid fish, where adaptation of the mouth and different structures to different food resources cause diversification and speciation. And what I show you here are different mouth morphologies and the morphology of the pharyngeal jaw, a second pair of jaws that cichlid fish have in the pharyngeal region. So we have plenty of examples showing that habitat and food resources adaptation cause diversification and speciation. And what we want to explore is how species interactions could also induce differentiation, speciation, or contribute to that. So the idea here is that if you live in different environments, you're exposed to different parasites, and this will induce changes in the immune system, and this eventually could be influencing mate choice and diversification and ultimately speciation. And this does work if you have a magic trait, as MHC genes are, the major histocompatibility complex genes of the adaptive immune system that they respond to different parasite communities, but they also have an influence in mate choice that has been proven in different organisms, in some fish examples as well. So if you are exposed to different parasites, your immune system changes. The MHC repertoire has been shown in different organisms to change, and if this has a link to mate choice, then this mechanism could complete speciation. So I work on cichlid fish. You have already seen a couple of fish in my images, and these are a good model to study speciation because they speciate a lot in different places of the world. And here's some quotes, but probably you have heard some talks about cichlids today here. Cichlids are particularly diverse in East Africa, but they also exist in America. In South America, the large American rivers are full of cichlids. We have seen here some rising nice models in South America, but they also exist in Central America. This is a very cool part of America because it has not only large rivers, but lakes, and cichlids have shown to diversify particularly in lakes. And what you have here is two large lakes that will act as a continent, that they are connected by rivers and full of fish and different organisms. And you see around there on the left, a bunch of crater lakes derived from volcanic activity. When they originate, they're empty, and they have been colonized by different organisms, and upon colonization, diversification happens. So this is a very nice continent island model. So this is the aspect of these lakes, pretty isolated, not so little, but very isolated, hard to reach. And one species of cichlid has reached all these lakes, the Midas cichlid, and they have diversified in allopatry, and there's enough genetic data that show that these lakes contain genetically different populations. But what is most interesting is what has happened inside each of these lakes, because diversification has also occurred, and genetic and genomic data prove that each units within these lakes are also genetically different. So there's a speciation going on here. But what is most interesting is what are the main axes of diversification in this system, and we see very nicely the typical benthic limnetic adaptation in many of these lakes, given there's enough depth. We also see some different trophic adaptations. As I told you before, habitat and food really play a role in here. So what we did was to study the parasites of this system, and we found plenty, and I show you this because this was a lot of work done by Anna. Mainly, so we focused on macroparasites. We found mostly trematodes, nematodes, acanthocephalans,
Copepods and monogeneans. These are the main groups that we found there. This was humongous work. We found a huge diversity of parasites, but what we found is that many were rare and few were common, and we found 8 taxa that were basically in every single fish we sampled. And there are 2 ectoparasites, 6 endoparasites. And what we saw is that the main relevance apparently here is the abundance of these parasites. Not absence, presence, but more how many of those you have. Because if you're in one of these lakes, you will eventually get basically all of these. So I show you here an example of 2 lakes where parallel evolution appears to have occurred, where we have benthic and limnetic fish, but we have a third set of species that have colonized the deeper benthic region. And we have analyzed the parasites in this system, and we see that consistently limnetic pelagic fish have more endoparasites. Here you have an example of some nematodes in the guts, and in the 2 legs we have a nice parallel infection of these endoparasites. If we consider all parasites, all these 8 parasites together, again, the limnetic fish have more parasites. You see there that they don't necessarily have the same parasites, but they do have more parasites. And this is a third lake where we find the same pattern. Limnetic here, we don't have deep benthic, but we have limnetic having more endoparasites. And what I show you here is that actually ectoparasites are more abundant in benthic fish. And this is consistent in these 2 parallel lakes. And again, in this third lake, missing the deep benthic species, we also have more ectoparasites on the benthic species. And I bring this here because in this third lake, we are finding that one of these species has a humongous spleen, as you see on the right corner. And well, we believe this is related with this parasite infection, but just to discuss that with anybody interested later. So we have done gene expression analysis on the spleen, which is a major immunological organ. And what I show you here is just that in the benthic area, most genetic differentiation appears to be happening, and towards the deeper region, you see fewer differentially expressed genes. But what is most interesting, and this is just a compilation of this data, but what is most interesting is that when we compare limnetic fish with any other one, is where we see in yellow that most immune genes appear to be overexpressed. So there's an increase of response, immune response in limnetic fish that is consistent. The same if we compare within these lakes, we have most things happening on the benthic area in terms of differential gene expression, but we have most immune response whenever we compare the limnetic with anybody else. But we were particularly interested in major histocompatibility complex because this is the major agent of the adaptive immune system. And what we found here was huge diversity. So in cichlid fish, it has been described at least 17 copies. We have 13 copies in this system, and we found good news, just a huge repertoire to adapt to different systems here. What the colors here show that the different alleles of MHC group in different functional groups, and we believe that will not be specific alleles, but more groups of alleles that will have a function. We see that the MHC repertoire is different in the different lakes. And the colors that I have highlighted here with names are these 3 lakes I have been mentioning. So there is probably, well, we don't know yet if it is allopatry or adaptation. We have different repertoire of MHC alleles in the different lakes, but we also have a different repertoire of MHC alleles in the different species within the different lakes. And so this was so for the 2 parallel examples that I show you, but also in this third lake where we have this huge spleen going on. So we would really close the circle if we would be able to prove that MHC is involved in mate choice, but I don't have this data yet. The approach that we are using here, because I don't have the facilities to do experiments, has been to collect breeding pairs and type for the MHC to see if they have more similar MHC, but I don't have this data yet. So in conclusion, it looks like the limnetic region is imposing a very strong pressure in terms of parasites, and that fish are responding, the immune system of these fish is responding more strongly. And so this mechanism where parasites associated with trophic resources and environments could be initiating, complementing, helping this speciation process. So with this, I want to thank the funding agencies, collaborators, and if there's time, I'll take questions.
Thank you very much. Yeah, there is time for questions, plenty of time. Yeah, Dan in the back.
Thanks so much. It's very nice to see this coming together. So cool work. I'm curious about MHC and to what extent you see copy number variation in competitiveness.
You mean copy variation within this system in the different species? So we don't have any evidence that this happens. We haven't looked carefully enough. What we see is that different populations have more or less average, so overall number of alleles. So we have populations that are isolated where their repertoire is smaller, but the thing is we don't know yet where in the genome are all the different copies of this MHC. And the genomes, even if they are high quality, they are not providing this information so far. And so now that there are some long reads, so some genomes with long reads, we are trying that. But we have an issue with locating all these diversity of alleles. But what we do see is that we have done an analysis combining MHC alleles from the cichlids in the neotropics and those in Africa, and they combine very well. So it doesn't really look like there's a big difference in at least those, so the number of copies of these genes.
Any other? Yeah, go ahead, in the back.
Do you think there would be a relationship between the number of variants in MHCs and the diversity of parasites rather than the abundance of parasites? You'd imagine you just need like different levels of expression for abundance, but variants.
Yeah, so yes, it could be. So right now, so we're studying number of alleles, and we're studying expression of those alleles, and now we are combining those alleles that are most expressed, which are not those that are most abundant, with the parasites, where this is work that is ongoing. But I think the theory says that a few MHC alleles could respond to a wide range of parasites. But we have these two data sets now, but we haven't put them together yet.
Any further questions? Then we'll thank Marta once more.